The endocardium is a thin layer consisting of an endothelium and some underlying connective tissue. It varies in thickness from place to place and is normally much more developed in the atrium than in the ventricle. Endothelium is a simple squamous epithelium with elongated or flattened nuclei. It is the special name given to the epithelial lining of blood vessels. The epicardium is a fairly thick layer of loose connective tissue. It has a large number of fat cells, making it stain palely, as we saw earlier in the gross anatomy image. In fact, fat cells are a dead giveaway for distinguishing the epicardium from the endocardium. A single layer of mesothelial cells covers the free surface of the epicardium. Mesothelium of the epicardium is a simple cuboidal epithelium with rounded nuclei. Now, the main layer and thickest layer is the myocardium, which corresponds to the tunica media of an artery. It is a thick band of darkly staining cardiac muscle cells, or cardiomyocytes. You may be able to see that cardiac muscle of the myocardium is sectioned in various orientations, but mostly transversely in this section and stained with hematoxylin and eosin. Note how irregular the individual muscle cells are in size and shape. This is in part because cardiac muscle cells branch. The myofibrils are readily apparent as red profiles packing the cell's cytoplasm. They are ribbon-shaped instead of rods, as is the case for skeletal muscle. Cardiac muscle cell nuclei are large, pale, and, as you see here, are located in the middle of the cells. Quite a few other smaller nuclei lie in the connective tissue spaces between the muscle cells. So most of these will belong to endothelial cells, lining small blood capillaries, and also fibroblasts, responsible for making the connective tissue support for the cardiac muscle. The great abundance of capillaries outnumber cardiac fibres. They are these tiny little circular profiles you can see very closely associated with the cardiac muscle tissue. Thicker seams of connective tissue, exaggerated here due to tissue shrinkage during processing, package cardiac muscle into fascicles or bundles and provide a pathway for arterioles and venules.